Hi, this is Dr. Dan Guerra coming to you from Authentic Biochemistry Studios in the Inland Pacific Northwest. This is the 13th of May, and what we're going to do is talk about lipid metabolism. Gee whiz, doesn't that sound like a lot of fun? I know it does, because once people learn about lipid metabolism, they really quickly understand that it's something, if they hadn't known much about it previously, they wonder where they could have ever walked out the door in the morning and taken a walk without knowing about it. And those of us that know a little bit more, because we're lipid biochemists, are only grateful when we learn yet more about lipid metabolism, because it is an eternal amount of information that will take an infinite amount of time to be able to event into our minds. So with that, supercharged positive reinforcement, let's get into this. Again, it's the 13th of May, 2020. This is an Authentic Biochemistry video lecture, so it's something novel and wonderful. And I'm Dr. Dan Guerra, as you are probably aware, because you've been watching these for a long time, if you have. And so let's just get going with this. All right. Now, this is going to be a large arc of lectures, and like I do always, I'm going to make it very relevant to medicine. Particularly at the very beginning of this one, I'm going to make you think back on just the last couple of lectures we've done talking about ketone bodies. This is going to be another cut into that area, and in fact, that's what gave me the inspiration to do a larger lipid discussion. Now, I'm not going to just stick with that, of course. I'm going to also talk about carbohydrates and proteins and amino acids and, of course, nucleic acids, because if we didn't do that, God forbid, right? But seriously, I want to focus in on lipids as much as I can because they are so important for human health. So let's cover some really general descriptions here. What's the role of the liver and the adipose tissue in digestive lipid metabolism? Okay, this is something that probably many people don't think about. The liver does many things, but from this point of view, I want you to consider the following. It synthesizes and secretes bile salts. Don't worry, we'll show you those in a moment. It synthesizes, secretes, and receives lipoproteins. It synthesizes new lipids, that is usually in the form of membrane lipids and also triglycerols, from non-lipid precursors, and transiently stores them, usually as either triglycerol or some kind of phospholipid. Now, adipose tissue, the other major player here that I want you to think about in the human body. Of course, every region of the body does lipid metabolism, but these are two main organs I want you to consider for right now. It absorbs triglycerol and cholesterol, usually as cholesterol esters, from chylomicrons. Now, those are circulating lipoproteins that come originally from the small intestine. And the adipose tissue absorbs these lipids, because both TAG and cholesterol are lipids, of course, through lipoprotein lipase. It's an enzyme associated with adipose tissue. And what does adipose tissue do? We know this because this is visceral fat, right? This is depot fat. This is what you notice as a tire around somebody's body as they get older if they don't get enough exercise and they eat too much. That sedentary lifestyle that then results in obesity. Of course, you can put adipose on all over the body, and there is a dichotomy between male and female about where that lipid accumulates, and I don't think I need to go there right now. At any rate, I think I've told you many times that we're oleaginous organisms. That means we store depot fat. I've explained to you why that is, because we don't need water to be able to store this highly condensed, reduced form of carbon. And the more reduced carbon is, the more energy you can derive of it, because then when you oxidize it, you drive away electrons. When those electrons are driven back into something else, like an electron acceptor, and protons are pumped across the inner mitochondrial membrane, for example, you can generate ATP. That's basically one or two sentences. I didn't actually count them as I was speaking. for the entire electron transport chain, oxidative phosphorylation, synthesis of ATP in the mitochondria. Don't worry, we'll get back to this many times. So adipose isn't just something that is like to be hidden or to not be discussed. Adipose is a very vital organ in the human body because it allows you to store carbon, right? It also is a basic network for signaling to the rest of the body, including, yes, the central nervous system, the brain. So it's a major organ system for dietary and for the BMI set point. Now that's another important feature here, okay, that I want you to consider and think about a lot. The BMI is often considered to be the important critical factor involved in whether or not a person is obese, right? Unfortunately, it's not always the case because it's a ratio and proportion sort of calculation. And that means that people that are very muscular might have a high BMI, but if you're not measuring depot fat, which is usually most of the problem with obesity, you may be talking to a person that's extremely healthy. I think you could think of a lot of like athletes in your family, or maybe you're one, or you're still active as an athlete. You know that you may have a reasonably high BMI, which puts you in the, gee, you're overweight, and you know that you're not overweight, right? And it's not just because you like to have a good self-image, it's because you're probably not relative to having some kind of disease state like obesity. Yet we can have people that do have a lot of lipid in their body that are not actually obese either. So it's not a clear association with just the amount of depot fat and whether or not you are technically obese, right? So body mass index has been used and I think largely abused many, many, many years now. And so people have gone to measuring, depot fat. That's a better indication of whether or not you could be leading down an unhealthy lifestyle or an unhealthy system, but it's not necessarily the case either. That's what I just tried to explain to you. We're not talking here today about that, right? That's more general nutrition, biochemistry of nutrition, and we can do that some other time, but not today. So this diagram here, I forget which textbook I got it from, just basically shows you digestion. You take in dietary lipids in the form of cholesterol esters, phospholipids, and triacylglycerol. Some of these can go unchanged and go directly into the digestive process. Most of those lipids and some short medium chain fatty acids end up in being emulsified by bile salts. Now these are, of course, a product of steroidogenesis pathways. So that emulsifies, and with pancreatic enzymes, which are generated from the exocrine aspect of that organ, the pancreas, you degrade dietary lipids. And so once you get into the small intestine, the primary products of this early digestive process are free fatty acids, 2-monoacylglycerol, where there's a fatty acid on just the second position of a glycerol backbone, because those are common lipids to consume. But the most, of course, is triacylglycerol. And then cholesterol itself, right? And usually in the form of cholesterol ester. You also have remaining fragments of phospholipids, right? And those get reesterified. That means they get reassembled according to position on that glycerol backbone and indeed on a sphingosine backbone. And then they are sent back into the system to end up into the bloodstream, back to the liver, ultimately associated with a lipoprotein called the chylomicron. It's the first lipoprotein we're going to encounter in digestion, the chylomicron. The chylomicron does get taken up by the liver because there's receptors for it, but it also can be, the lipids on it can also be taken up by all of the periphery, including muscle tissue. So I want you to understand, and of course adipose tissue, I want you to understand that the lipids that are associated with the very first lipoprotein can be delivered and then hauled off of that lipoprotein and then stored in various organs in the body, including skeletal muscle, cardiac muscle, and of course depot fat. Okay. All right. So I want you to keep in mind a couple of other things. Free cholesterol is very seldom found on lipoproteins, usually in the form of cholesterol esters. cholesterol esters meet up with, as they say, an adipose tissue, an enzyme called cholesterol esterase. We've talked about these in previous lectures. I'm not going to do it right now. What that does is remove the fatty acid from cholesterol. So you actually get two things when you take up cholesterol from the diet. You get an esterified fatty acid, okay, it's going to be an oxygen ester, that gets removed when it gets stored in depot fat. And then you also get the cholesterol molecule itself. Now, as it turns out, you don't have to consume cholesterol in the diet. You require cholesterol, of course. Remember all the steroidogenesis we talked about in previous lectures last week, to make all of the really important, like corticosteroids, mineral corticoids, as well as, of course, the sex hormones, right? Testosterone and estrogen as examples. But you also need cholesterol in the membrane, really significant in the membrane, just for how the biomembrane functions, right? So the body doesn't allow for you to need that as an essential. So you synthesize cholesterol de novo, and we talked about that via the HMG-CoA reductase pathway, right? The whole mevalonic acid pathway, building up basically from acetyl-CoA, just like fatty acids are, all the way up to the cholesterol backbone, the cyclopentane-phenanthrene ring structure that ends up being what that steroid nucleus looks like, which is looking at there. Now, the other thing to keep in mind, too, is that you get a lot of reactions with phospholipids. This is phosphatidylcholine. Notice that choline is a trimethylethanolamine structure, right? It originally came from serine. That's where that so-called head group comes from. And it's a phosphate ester, right, on that 3 position of that particular phospholipid. And you can remove fatty acids from it. And this particular reaction is showing you take off these two, and now you have a glycerol phosphorylcholine backbone, and you have no fatty acids. What I'm telling you is more often you get lipases removing fatty acids from either the 1 or 2 position. And then there are phospholipases that remove that head group. We talked about those two. It's a really important signaling. Triacylglycerol is a little bit more straightforward because you're not dealing with a very specific alcohol, for example, 3 position glycerol. This is just showing you glycerolipids. Remember, sphingolipids are just as significant, although they're less of a component in the diet. But triacylglycerol broke down again by lipases. There's the pancreatic lipase functioning correctly, removing fatty acids, making this 2-monoacylglycerol I told you about that end up being reformed in the intestine, packaged in the chylomicron, sent into circulation, changed and altered in the liver, and then the liver becomes a major organ where all of the lipoproteins you've heard of, like the low-density lipoprotein and the intermediate density lipoprotein and the VLDL, it's very low-density lipoprotein, are actually synthesized and secreted. I also told you about how chylomicrons need apolipoproteins, and all of the lipoproteins do. Apolipoproteins means simply before the lipoprotein's made. Remember, these are non-covalent associations of lipids with a series of proteins, and these apolipoproteins also need de novo synthesis. And in the small intestine, those apolipoproteins are synthesized according to the amount of lipid in a given meal. And when you digest a lipid meal with protein, the protein is broken down to individual amino acids via proteases in the small intestine, not for utilization as energy or even absorption, but for those amino acids to then be used, in a translational process that is taking RNA and turning it back into protein. And now those proteins are apolipoproteins. They're going to assemble as chylomicrons and haul off the lipid. So your body is really good at manufacturing that. And I've also mentioned to you that if you have animal proteins in the diet, that's good, because animal proteins are enriched with the correct mole percent of amino acids that you're going to need to make mammalian chylomicrons. If you're consuming only plant proteins, for example, you could have a deficiency in certain amino acids. And I don't just mean essentials, because you're degrading these proteins in the small intestine, so you don't have, that's a rarefied place right after a meal. You may not have the appropriate stoichiometry and mole percent of all the given common 20 amino acids to be able to build that apolipoprotein structure to be able to give you significant chylomicron assembly, and therefore you get poorer lipid digestion and assimilation. And that can give you problems with small intestine and with lipid accumulation there. I want you to keep that in mind. That is a nutritional tidbit, but I don't want to walk by it. Now, three lectures ago, we were talking about the fact that white matter lipids can act as a ketogenic source of basically bioenergetic ketone bodies for the neuron. And we talked about, the astrocyte is involved in making these, excuse me, let's get this, let me just use this, I guess. The astrocyte's involved in making the ketone bodies, and the ketone bodies are transferred via a transfer protein that's on the membrane of the astrocyte. A monocarboxylic acid transport is what MCT is. It enters the ketone body, either acetoacetate or beta-hydroxybutyrate, enters into the neuron, and then that is a source of carbon for the neuron. And I told you in many lectures past that in one of the hallmarks of prodromal Alzheimer's disease is a shifting of using glucose as the major carbon source in the neuron to ketone bodies. And so you think, well, does that mean ketone bodies that I'm synthesizing because I have a high ketogenic diet? No, that's not what I'm talking about right here. I'm talking about ketone bodies being synthesized in the white matter, right, in the astrocyte, which is one of the glial cells associated with the neuron in the central nervous system. And if you start generating ketone bodies as a major carbon source for neurons, particularly in places like the hippocampus, as you get older, because of problems of glucose transport or glucose uptake and glucose utilization, maybe because of insulin resistance or the lack of insulin responsive genes downstream from insulin binding, or maybe the GLUT transporters. There's all kinds of malfunctions that occur as you get older, that your brain relies more and more on ketone bodies. So it's a different thing than talk about a ketogenic diet. I want you to think that ketones are bad, because the ketogenic diet can be very good for people because it means you're eating more lipid, which you should, and less carbohydrate. The point I'm making here, though, is that the excessive amount of ketone bodies that might be made in the astrocyte, translocated via that monocarboxylic acid transporter to the neuron, and then the faithful utilization of those ketone bodies to generate, now acetyl-CoA, and that acetyl-CoA in the neuron, there it is finally, the acetyl-CoA in the neuron then, guess what? It gets used in the TCA cycle to make NADH, right? And the NADH ultimately gets oxidized in the electron transport chain, oxidative phosphorylation, they make ATP. You need a lot of ATP in the neuron, right? You know that, right? I mean, if anybody has ever listened to any of my lectures or ever listened to any physiology lectures about humans, the brain needs a lot of energy, and this is what we're talking about. Neurons need a lot of energy. It takes a lot of energy to move around neurotransmitters, to move around transcripts so they can be translated. You can make neurotransmitter, proteinaceous, or the ones that are generated from amino acids, for example, even lipids. So you need a lot of energy in the neuron, and so the neuron cannot wait for energy, and the astrocyte is there to be able to provide that carbon in the form of ketone bodies. So that's an important thing to keep in mind. Now, here's something I want you to think about. This paper published in Current Opinion, Clinical Nutrition and Metabolic Care. Not a journal I often look at, but this one caught my attention. You see the direct citation there. Essential mitochondrial succinyl-CoA oxoacid transferase, an enzyme we've talked about. This is a ketone body utilizing enzyme. It's also known as SCOT. It converts acetoacetate to acetyl-CoA. Okay. It's absent in hepatocytes, and that's presumably it blocks the use of ketone bodies for energy in that organ. Remember, the liver is a major source of ketone bodies, particularly during what? During fasting or during starvation or during low glucose or low carbohydrate diet. Right? So histone lysine acetylation, now listen, following here, diminishes the chromatin electrostatic interaction between double-stranded DNA and the lysine-rich nucleosomes, which is how chromatin is packaged, thus remodeling, when you have that acetylation, right, on those histones, so lysine residue on those histones, to euchromatin from heterochromatin. And you open up, that DNA is then open to an increased probability of RNA polymerase and all the single-stranded binding proteins to be able to get into that remodeled chromatin and allow you to get transcription. Okay? So it aids in transcription, acetylation does. Methylation, now this is another way of altering lysine residues that are in histones that are associated with chromatin in the nucleus, right? Methylation is more complicated because there is also lysine ubiquitinylation, okay? So methylation itself is complicated in that if you methylate the DNA, do you make more heterochromatin? Yeah, kind of. It gets more condensed because remember, that's very hydrophobic, that methyl group, whereas acetate is very hydrophilic. Therefore, you can understand that it would expand the DNA. But it depends on where the methylation is. And also remember, you get methylation what? On the DNA itself. Remember the CpG islands. So a lot of other things you can think about with methylation. Acetylation is more straightforward. But besides methylation, besides acetylation, you get lysine ubiquitinylation, propionylation, butyrylation, 2-hydroxyisobutyrylation, succinylation, that's a very important one, malonylation, that's another important one, glutarylation, hydroxymethylglutarylation, crotinylation, and even beta-hydroxybutyrylation, beta-hydroxybutyrate, right? Ketone bodies. So you get the idea here, okay, that you can have modifications, covalent modifications of histones. So we just talked about, ketone bodies being used, directly for bioenergetics. Absolutely the case. Now I'm telling you that you can make a lot of these acyl groups, right? Some of these acyl groups are directly from ketone bodies, or they're from partial degradation along the pathway of beta oxidation of fatty acids. Remember, fatty acids can't live free and easy because they are what? Insoluble in aqueous. So they're bound up as an oxygen ester or as an amide linkage to something like a protein, or even what? As a thioester, coenzyme A. That puts them in biological activity. That's where that carbonyl ends up, right? The carbonyl of the fatty acid just hanging out there free because the fatty acid would be basically insoluble and therefore inert in bioenergetics and all of the rest of cellular metabolism. So I want you to keep all of that in mind. So this is just the pathway itself. You start off with fatty acyl-CoA, beta oxidizing down, ultimately to acetyl-CoA, but then acetoacetyl-CoA, because now I'm showing you the ketogenic pathway. You have the HMG-CoA synthase, which takes on one more acetyl-CoA. So now you have in the mitochondria, hydroxymethylglutaril-CoA. Then you have a lyase reaction, which removes the acetyl-CoA, and that allows you now to just have acetoacetate. You're no longer a thioester. So you need the thioester in metabolic biosynthesis degradation. You don't need it now that you've made a water-soluble compound, acetoacetate. Acetoacetate can also be used to synthesize, right, via an oxidation to beta-hydroxybutyrate, right? A reduction. So acetoacetate can be converted to beta-hydroxybutyrate, and it's the acetoacetate and the beta-hydroxybutyrate, right? Those are your two major ketone bodies. The enzyme that does that is actually in the mitochondrial membrane, the outer mitochondrial membrane. Right? So now once utilization occurs, right, one of the simplest ways to utilize it is with succinyl-CoA. Now if you're in the mitochondria, there's plenty of succinyl-CoA because of the TCA cycle, right? And so because there's a sufficient amount of succinyl-CoA, this enzyme here, SCOT, the one I just told you about, will take succinyl-CoA and make free succinate, which is perfect because it goes right through the TCA cycle. But now you've re-esterified the acetoacetate to acetoacetyl-CoA. Then there's an enzyme which will utilize more CoA to give you two acetyl-CoAs, right? So acetoacetyl-CoA only has one coenzyme A. When you add coenzyme A, CoAsh is a reduced form with the thiolase reaction. That's what carries out that cleavage of that bond because it's a thiolytic reaction, thiolytic cleavage. You then get two acetyl-CoAs. You run those through the TCA cycle after binding to oxaloacetic acid. Because NADH levels are low, you run all that NADH up through all the dehydrogenases in the TCA cycle, and then you make plenty of NADH to be able to run through the electron transport chain, right, through the four major complexes, ultimately get the proton pumping through the fifth complex, the proton pumping ATPase, and make ATP. Now, Okay, so hopefully that's what's saying this over here. Beta-hydroxybutyrate, acetoacetate, primary circulating ketone bodies. They are increased in ketosis. We've heard of ketosis and ketoacidosis. In synthesis and circulation during fasting, starvation, stress, inflammation, and low carbohydrate diet. All of this is going to increase. Now, that's the ketogenic process, and that's good. Now, what I just showed you in the brain is different. That's in situ metabolism in the brain, which can occur as you age because of a problem with glucose uptake and transport, right? Remember what I was saying. And we don't store a lot of glycogen in the brain, but we can store some lipid, not too much, but we are really efficient in making ketone bodies and astrocytes. And that's the new ripple, right? All right, so diabetic ketoacidosis is paradigmatic. You know that. You've heard about type 1 diabetes and having a lot of ketone. So beta-hydroxybutyrate and acetoacetate also serve to control gene expression epigenetically. This is the new cool thing. Right? This is yet the third new cool thing. And that's via control over histone lysine acetylation. Wow. So that's what I showed you over here. So this diagram here I took from one paper, and then I kind of modified it, showing you that there is a histone lysine canonical acetylation, and you get selective chromatin remodeling and alteration of transcription. Okay? That's what I'm trying to show you here. Really an important feature. So that means if you're making a lot of ketone body in the astrocyte, and then you're utilizing the ketone body in the neuron, you're building up a lot of acetyl-CoA that way. That's fine. You need the ATP. But one of the toxicities that we talked about way back, probably a month or two ago, was some of that acetyl-CoA can be used to rebuild palmitate, and palmityl-CoA can be used to make ceramide, and ceramide can be associated with neurodegeneration. Remember that? Right. So that's one issue. What's the other issue? Now I'm telling you that some of the acetate in the form of acetyl-CoA being used as a precursor substrate can now be used to covalently modify histones. Now, didn't I just tell you that that means more transcription? Yes. But all of those other covalent modifications of all those other acyl-CoAs as substrates, like beta-hydroxybutyryl, for example, protenyl, some of that, or even excessive acetylation, not just of the histones, but of proteins in the mitochondria that are very active in the central nervous system, obviously, it's very aerobic. If you get acetylation in particular of a lot of mitochondrial proteins, which apparently happens in Alzheimer's disease and happens too in obese diabetic patients, that corrupts the activity of the proteins that become hyperacetylated, and that contributes to the increase in reactive oxygen species, which then in combination with the ceramide genesis can result in neurodegeneration. Hence the significance to CNS neurodegeneration such as Alzheimer's, Parkinson's, and other kinds of degradative processes, even like MS. We talked about these things, but this is a new ripple. This is showing you that epigenetic changes, right? Because these are going to be epigenetic on histones. They're going to alter gene expression. That's one thing. They could become corrupted with excessive amount of acetylation. It means there might be transcription of genes which normally are not transcribed in that region of a neuron, or that region of the brain where that bed of neurons are, like in the hippocampus or the hypothalamus or the pituitary or prefrontal cortex, somewhere else, right? The amygdala. And if that's going on, that's going to alter not just gene expression, but then the proteins that are ultimately synthesized after transcription and then translation can completely alter bioenergetics, signaling, and fate of cells in the central nervous system. So that's the point we're getting at here. So covalent modification, protein nucleic acid, are a few of the epigenetic mechanisms that are sensitive to real-time environmental stimuli that subsequently modify transcription patterns as a response to that stimulus. Okay, that's the first take-home proposition. The term environment, the way I use it, simply means phenomena that must be tracked from both external and internal alterations in set point homeostasis. That means things that are happening inside a cell just because of normal modifications, turnover, the vagaries of a cell and its cell cycle, but also any stress phenomenon or any alterations in nutrition or change in bioenergetics or in signaling or in the synthesis, for example, of pro-inflammatory cytokines after an induction associated with, say, an innate immune response from toll-like receptors, right? So these phenomena are associated with danger signals, which we talked about, such as pathogen recognition receptor-mediated sequelae, nutritional signals via the canonical endocrine pathways we've been talking about, right? Think about the adipokines, secondary agency from the immune system, including the activation of innate immune responses, something I just mentioned, I just flew it up, cytokine, chemokine growth factor, antibody secretion from that system, and ultimately, after absorption and uptake of lymphocytes to the blood-brain barrier, lymphocyte-mediated inflammatory, this is if it's in the CNS, could be in any tissue bed, inflammatory responses and the recovery to a set point. All of that can now be altered because of the phenomena I just explained to you. Right? At the clinical level, epigenetic regulation of gene expression helps to maintain security and maintenance, terms I've introduced into the discussion here. Security and maintenance of the biological system as read through physiological adaptation, which happens all the time. It doesn't have to be pathophysiological, although you can put that in parentheses ahead of that word, depending on what's going on in terms of hyperinflammation, for example, right? Or the perhaps infection from some infectious agent. So bioactivated carbonyl carbon, which is what we're talking about here, is a common source of epigenetic covalent modification, whereas phosphate ester addition, another covalent modification we talk about all the time, kinase cascades, and removal of that phosphate is more the realm of immediate course correction of kinetic parameters, for example, Kms and Vmaxes, and subcellular trafficking, let's say, of endosomal fragments or of membrane fragments like membrane rafts and whatnot, right? That could be affected by phosphorylation. But this longer-term effect of acetylation or any of those other carbonyl-associated acylations can not just modify immediate responses, but slightly longer-term responses. Right? Again, that can then induce a whole chronic picture to this story, right? Chronic pathophysiology, for example. All right. Now, this is a really interesting thing. Both the pH and the acetyl-CoA concentration of the mitochondrial matrix, if you're generating a lot of acetyl-CoA, let's say from ketone body utilization, let's say in the neuron, which is what I just showed you in the aging brain or in the AD brain or the PD brain, they're elevated compared to lysosome, cytosol, and nucleus. They both are when you're using a lot of ketone bodies in the brain, and the pH of mitochondria is always higher than lysosome, which is very acidic, cytosol, which is more acidic, and nucleus, which is again near neutrality. So a pH 8 in the mitosol, remember that's the intermitochondrial matrix, versus pH 7.2 usually in those other subcellular regions. And popping up to 0.1 to 1.5 millimolar concentrations of acetyl-CoA in the mitochondria, because you're generating a lot of it. Beta oxidation sensu stricto, or from this utilization via the thiolase reaction of incoming ketone bodies, which is the brain is going to be serviced by these various nutritional choices, as well as by aging, pathology, and stresses I've already described. So here is now a new ripple. Yes, and many ripples today. Non-enzymatic acetylation is analogous to lysine modifications like glycation and carbamylation, right? Now this comes from a paper which I pulled out from the Journal of Biological Chemistry 2013. Of course, fantastically important journal, JBC. I hear that all new papers coming out in 2021 and ongoing in JBC and in JLR and the Journal of Cellular Biochemistry, I think that's the third one, which are all owned by this parent company, are going to be free. That is really cool. I just hope that the quality remains the same. At any rate, what is this showing you? When you have a higher pH and you have incoming fatty acids for oxidation, it doesn't show here, but let's add ketone bodies in the neuron. I should modify this because it's directly from the paper, because I will modify it myself. Glucose coming in, or amino acids. Ultimately, all that's going to generate acetyl-CoA. Glucose via what? Pyruvate dehydrogenase, last reaction of glycolysis. Fatty acid via beta oxidation. Amino acids after transamination, and then production of alpha keto acids, and then ultimately, of course, acetyl-CoA. Amino acids can be converted to acetyl-CoA. High concentration, showing you here, like we're talking about, mitochondria, and then this high pH. Well, you can get these reactions. Look at this. Endogenous non-enzymatic lysine modifications like N-glycation can occur. Okay? And notice what's going on here, right? You're getting, the mechanism involves the primary amine because of this high pH. See, it's deprotonated here, so you got these free electrons here. It performs a nucleophilic attack on the carbonyl carbon of the endogenous metabolite. So you can get N-glycation. You can get N-carbamylation. We talked about both of these in previous lectures, now that I'm reflecting on it. And here for today, N-acetylation. And again, they have a question mark there because when they published this paper saying, well, we're not going to say this, but this is what they discovered. And plenty of papers since 2013, October, so almost 7 years later, have buttressed this response. So this is non-enzymatic acetylation of proteins. I just told you that excessive acetylation of protein, even if it's enzymatic, can lead to disruption, right? Dysregulation of pathways, ultimately leading to a pathological status, which can generate something like an immune response from an innate immune response via danger signaling, right? And then, or that can ultimately then lead to an inflammation, which can cause ultimately neurodegeneration. So now you get the idea, non-enzymatic means. And down here, this is from them, many coester-mediated acylations may occur non-enzymatically if the subcellular compartment is chemically poised, as in the alkaline pH in the mitosome. So whenever you raise the pH, if you have plenty of this carbonyl as an ester, right? Or if you have plenty of this carbohydrate or carbamylates, right, which come from a whole host of reactions in amino acid metabolism, I can tell you, then you can get a non-enzymatic alteration of lysine residues. If those lysine residues are in enzymes, shift bases, or you're making these amide linkages and whatnot, or if they're in histones, you're going to greatly, and there are, of course, what? There's also transcription, right, in the mitochondrial genome, right? Because you have a mitochondrial genome. So this plays a lot of different potential pathophysiological inceptions, okay, in the cell. If you go through all these papers, you'll learn this. So this comes directly from that paper, JVC 2013, October 4th, great paper. Excessive protein acetylation in the mitochondria can cause dysfunction of bioenergetics, leading to reactive oxygen species accumulation and the potential for various forms of apoptosis. You know, the kind that are the most dangerous, like ferroptosis and necrotosis, right? This can occur in obese patients, especially in organs like the liver, which we started off the liver this morning, where excess fatty oxidation is certainly going to occur. And remember, the liver doesn't use ketone bodies, so they go out in circulation. Also occurs during stress, where rampant glycolysis presents aerobically cancer or hyperimmunity, right? Here's another place where you're going to be able to build acetyl-CoA, rampant aerobic glycolysis, which is kind of a hallmark, right? Randall cycle stuff associated with oncogenic events like tumorigenesis, right? Also profound, this can also occur during muscle wasting, right? Like cancer cachexia, or excess protein in the diet. These bodybuilders that consume a lot of protein, you get a lot of nitrogen, nitrogen itself, ammoniacal. Ammonia-based nitrogen is also dangerous to the body, right? As well as the degradation of these amino acids with acetyl-CoA, now I'm telling you. That leads to amino acid degradation, ultimately excess acetyl-CoA production, which is not good for these reasons, right? So the next phase of this lecture is going to be talking about lipid absorption. I'm going to tell you, I'm going to discuss these things at some length, directly into the bloodstream, glycerol and short, medium-chain fatty acids, the lymphatic system for all the lipoproteins. My cells diffuse can also directly into intestinal cells. There's a reassembly of triacylglycerol in the small intestine, packed with the proteins. Those are the apolipoproteins, make chylomicrons. And at first it bypasses the liver so it can transfer directly from the diet those lipids, right, to the periphery. But then subsequent to that, you get chylomicron uptake in the liver and then the production of all these other lipoproteins. LDL and HDL we've heard before. I will mention these and I'm going to stop today. So LDL is the major lipoprotein transporter for hepatic triacylglycerol and cholesterol as cholesterol esters. Liver regulation of the gene expression for those apolipoproteins and receptor turnover is very well balanced, going all the way back to steroid-associated cleavage of proteins so that it modifies the expression of genes at the level of what? Transcription factors, right? We talked about that before. It's Browning-Goldstein type of work. High-density lipoproteins, that's LDL. There's a whole lot more to say about that, but that's just a nice little, short discussion of it. Of course, HDL, high-density lipoproteins, remove cholesterol from cells, carries cholesterol to the liver for recycling, and HDLs have been claimed to have anti-inflammatory properties, but it's not the HDL itself, it's what's associated with it. And it's actually also the mechanism by which the body uses HDL. LDL and HDL are not bad and good lipids. Don't ever say that because that's absurd. They are canonical lipoproteins that are in the body, and they serve absolutely, I'll even use the word, necessary functions. Right? So don't ever attach any kind of adjective to them, calling them good or bad. They are simply part of the repertoire of lipoproteins that allow you to digest lipids, utilize lipids, and basically so that the body is maintained, right? It's maintained for a long-term endurance and ultimately survival to the end. So I'm going to stop there and call back this section. And I'm going to leave you with a relatively short lecture, but I gave you a lot of things to think about, hopefully. Ketone bodies, the production of acetoacetate and beta-hydroxybutyrate, possibly other acyl-CoAs, ultimately, and occurring in the neurons can alter a lot of biosynthetic pathways, including bioenergetics, including signaling, which could lead potentially to a pathophysiological state. And then the general understanding of how excess lipids in just because of an obesogenic state is going to mean a lot of circulating fatty acid. Those fatty acids can be then utilized directly to produce potentially pro-inflammatory lipids like ceramide, but also even the fatty acids themselves can be used as covalent modifiers of histones and DNA, chromatin complexes, or as acetylaging agents on various mitochondrial proteins, which can alter things like the electron transport chain, oxidative phosphorylation, and a lot of other things that are happening in the mitochondria besides just bioenergetics, amino acid metabolism, further lipid metabolism, carbohydrate metabolism, nucleic acids metabolism, and all of those, if they're corrupted, can lead to, for example, programmed cell death, some of which can be the type that can induce an inflammatory response. So that's what I tried to get to you today. You see how I brought that in so that we're not just talking about lipid absorption. I'm bringing this in so that you understand there's a dynamic system at play well before you eat that meal high in lipid and high in protein and maybe low in carbohydrate. And every time you ingest a meal, a new infusion of signaling and regulatory processes have to occur. And that fasting, which is usually very good for the body, longer-term starvation, all of these dietary choices, low carbohydrate, high carbohydrate diets, are all going to affect at the chronic later level over time a tremendous potentially positive balance of carbonyl metabolism, or potentially a very dangerous pathogenic imbalance of carbonyl metabolism, all because of acetyl-CoA and its relative other thioesters used as covalent modifying agents for things like histone lysines and on lysine residues and other proteins. Okay, so with all of that, I'm going to end this lecture today on the 13th of May, 2020, with bye for now.